Okay, so our next speaker is Dr. Chia-Lin Wei from the Joint Genome Institute. She's a group lead there. Previously, she was with the Genome Institute of Singapore and also with large-scale biology. And she's been involved in the development of a lot of robust and innovative cloning technologies, and also played a leading role in pioneering the use of sequencing to interrogate genomes for functional units, epigenetic regulation, and also transcriptional networks. And her group is actively involved in the ENCODE and Phantom consortiums. Dr. Wei has published over 50 peer-reviewed articles. She holds over 20 international patents, and was in 2006 was one of the recipients of the Singapore National Science Award. So we're very privileged to have her here today. There we go.
All right.
Thank you very much.
First, I want to thank Kapa and then given me this chance to introduce my research and then JGI sequencing activity. So I'm going to incorporate a bit on the technology side with our biological interests and science, particularly on the biofuel side. So first, a few words about JGI. And I think at this meeting probably is not, JGI is probably heard in many different places. But it's just so you know, JGI is funded by Department of Energy as a genomic facility, particularly on the technology facility in support of all the DOE-based science missions. We're located just probably an hour and a half flying time away in the Northern California. We have over 260 employees and in the Walnut Creek area. The three particular scientific areas that JGI focus on are bioenergy, carbon cycling, and biogeochemistry. And how do we convert that to genomics? Is through a program, a list of biology programs that covers from metagenome, plant, fungal, and microbes. Recently, we add our capability to DNA synthesis. And then with this program to recruit good scientific projects, and the projects will be conducted by the infrastructure technology group. And the technology group covers from DNA sequencing, technology, computational analysis, and like I said, the recent addition is DNA synthesis. So my function is both responsible for technology development as well as running the high-throughput DNA sequencing. So if you have project with JGI, 99.9% of the possibility, I handle your project. So my group handle a project. So if you have any concern, questions, feel free to come back to me. I've met people from meetings that say, you guys should get fired because you don't do this. So I'm welcome for all criticisms, as well as a compliment if you have a good experience with JGI. But that's pretty much my role. But my group also run by scientific projects. One of the, and mostly beyond sequencing. So my interest beyond, once you have the genome sequence, my group's interested in understanding the functional elements. One of the interests we work on is from algae, and particularly because the biofuel. So I think if I introduced this slides probably a year ago, people are usually not in their head in the meeting rooms. But these days, because the oil price, gas price dropped significantly, probably you don't probably appreciate the needs for biofuel anymore. But I think most likely you will agree that this is an important area. And then we study biofuel because the biology, the plants, particularly have this magic power. Why magic power is, it has the magic power to convert sunlight through a very simple photosynthesis activity to organic matters. And most of organic matters are either in the ethanol field or some sort of esterified fatty acid. And these two type of categories are mostly commonly used in the source of biofuel. So in this organic matters between ethanol and esterified fatty acid, one of the sort of material or source comes to particular attention is the algae. So algae is different from what you thought. In your pound, when you have not keep a good watering system. It grew the sticky red, yellow, red, or green stuff. But it has this function, and when actually it produces lipid. And this is the staining. If you look at the yellow, it's some sort of neutrophil dye. It's staining specific the lipid body. And particularly this lipid body, you can actually go through a very simple chemical extraction process and directly actually used as a biofuel. So it is called a dropping oil because there's no need, like ethanol, to change the facility or gas station. It's just can be used directly. However, the problem for algae to produce this lipid, it has to go through stress. So the stress is when it has to go under some sort of lack of nutrition or high pH or some sort of chemical stress. That's the only time when they produce a lot of lipid. And in fact, when it does it efficiently, you can actually have a very high productivity rate. In a direct comparison, the amount of land and where it is to produce algal lipid compares to other sort of biofuel crop like corn or grass. You can use a lot less material or land, and also does not compete the land who produce a lot of agriculture or like some of the food products. So it's a very attractive source. So that's why we're actually interested to study what are the triggers for those lipid production in the absence of stress, because then you don't have to apply very drastic measurement to produce those lipids when the cell actually don't grow very well. So, and this is a model that we use is Chlamydomona. It is a small algae, has this two flagella. It moves in a pound, 120 megabase of genome. The gene space is well annotated, has about 15,000 gene model, and over 4,000 are curated. And then again, it's actually when under stress, it produces this lipid that convert the diacylglycerol to triacylglycerol. So to do this study, we actually trying to do a very comprehensive RNA-seq analysis because we don't know when or how this lipid trigger expressed. So we take the cell culture, grow into both the nitrogen starvation and sulfur starvation. As expected, when the cell under the stress, they don't grow. They stop growing, so the growth curve start getting flat compared to wild type. However, under those stress conditions, although the cell doesn't grow, it actually produces lots of lipids. So under this kind of over 48 hours time frame, we did a very, very high resolution time scale sampling of RNA-seq. And here I'm going to jump in a bit on the technology. So when you have this kind of experimental design, one immediately question is, how many RNA-seq I can do? Under the sunlight, if no limitation in your experiments, I want to do hundreds of RNA-seq because I want to make sure I do catch the dynamics. So in JGI, we have established a high-throughput RNA library automation, where we take the RNA-seq, standard RNA-seq protocol, but we put everything on the automation instrument. In that way, we can actually improve consistency and reduce cost, and then we can actually increase the throughput. Therefore, we can actually carry out hundreds of RNA-seq, and that's just a few days with one technician, and you get a very high reproducible result. And regarding the reagents, so we actually test the Kapa RNA-seq. So this is an experiment in collaboration with Kapa and taking the RNA input because we want to do a lot of RNA-seq. We also don't want to use a lot of material. So here we tried input starting from 10 nanogram, 100 nanogram to up to 1 microgram. And we do replicates of each input. And then we're subject to two types of RNA-seq analysis. One is the standard Kappa, one is their hyperprep. So the hyperprep has this one-tube chemistry. Because it's one tube, you don't have to go through individual cleanup or changing reactions. So it's higher yield, and also because it's one tube, it is easy to automate. It just fits our need because we really want to do a high throughput RNA-seq transcriptome analysis. So first of all, you can see high correlation between input amount starting from 10 nanogram, 100 nanogram, or 1 microgram. So this is not, this is really very little amount if you think about 10 nanogram from the starting material. And then not only within biological replicates, but between the hybrid kit and standard kit, and also between the 10 nanogram, 100 nanogram, 1,000 nanogram. So they all share overnight high correlation among them. Secondly, we also observed the kit generate a very uniform coverage between the genes. So this is the starting of the gene, this is in the gene. This is a coverage plot. So sometimes RNA-seq can give you very biased coverage. So you only cover like really the 3' end or partially in the gene region. So we only performed very standard sequence analysis, and then we actually observe very uniform coverage from the beginning of the gene, so the entire gene is well covered. Thirdly, just by the standard sequencing, we captured high number of transcripts. In fact, I think the captures number of transcripts from HyperKit is a little bit higher than the standard kit. Even they have probably less amount of material to start with. And the capture number of transcript didn't vary much from 10 nanogram of input to 1,000 nanogram input. So they probably because the HyperKit has a lot higher yield, and we can use much more. We can generate a lot more complexity library. Lastly, if we compare the expression RPKM among the range among all the samples, again, doesn't matter you start with 10 nanogram to 1,000 nanogram. The amount of RNA give you very comparable, reliable distribution RPKM, indicating the consistency of, yes?
Excuse me, is it all polyA selected?
Yes.
So you're starting with total RNA?
Yes, we start with total RNA.
Total RNA numbers and polyA selection.
Yes. So another things we add in our JGI process, we added the spiking. So the spiking is ERCC spiking that has a wider range of some sort of artificial RNA molecule, allowed us to determine counting accuracy, dynamic range, and then replicate the correlations. So if you have your project working with JGI, we actually can offer that information to you in case you want to do your own analysis to study your dynamic range using those spiking counts. So in conclusion, we think both kits, also one thing I didn't show here is both the kits have a high percentage of stranded information because we use stranded RNA-seq kit. So if you have a very high interest in bidirectional transcript or antisense transcript, that data will be available as well. So both kits offered very high specific strand information, and the HyperKit is able to capture more transcript overall. Probably because the one tube step process. Both methods equivalent measure the gene expression level down to 10 nanogram of starting material, and they share very high correlation. And the read coverage is very much uniform, and we don't have to detect a common like 3' bias in some of the RNA-seq analysis. And one thing to show that we have actually associated the chromatin state of those, again, we're trying to understand the functional chromatin state of those algae when they're under some sort of stress. So we actually can associate different gene promoter with different chromatin states with the gene expression level, indicating the RNA-seq does have a very good and sensitive and proportional readout for their gene activities. So taking the gene expression profile subject from nitrogen starvation and sulfur starvation, we look for co-regulated transcript under very early stage. So here we only take a time point from 0 minutes to 2 hours, very beginning, because we're looking for the early regulator, early step regulator. So we take the genes that actually 2 genes that looks like a transcription factor that seems to co-regulate in the first 2 hours under both stress condition. And then this is a example showing that a chromatin states showing a very drastic activation states under both nitrogen and sulfur starvation. And the RNA-seq also show a very rapid upregulation in between 1 to 2 hours under both nitrogen and the sulfur starvation. And one thing we went ahead to study their actually function by either doing gene perturbation or overexpression. So when we actually knock out the genes, the mutant shows the lipid accumulation phenotype has been largely affected. So the wild type with different stress condition start accumulating lipid, showing in the black bar, and the mutants in the red bar have deficiency in accumulate lipid, indicating these genes is required for this algae to accumulate lipid. And we measure the lipid from 2 independent dye activities, and then here is a visual of the cell. So on the wild type, when it's under starvation, accumulate lipid, showing in this red color. In the mutant, when this gene is knocked out, you have a greatly reduced the lipid content. It's shown this here. On the other hand, it's not only required, it's not only essential, it's required. It's sufficient. So when we just overexpress this transcription factor, we see a large increase of lipid content compared to wild type. So again, this is a regular fluorescent staining for the dye that capture the lipid, and this is the image when the cells undergoing this overexpression of this transcription factor, and then you have seen the cell actually become swollen. They become a lot of more fat. They have seen, you can see a lot of fat body. It's pretty sick in the shape. It's obese cells in that perspective. So this is some sort of give a dream potential. So we're hoping we can actually use this study to facilitate biofuels research, and the idea in one day you can actually produce this in a large quantity for really facilitate for biodiesel production. And then you can even use it for decoration in your backyard to see this. And it's not only beautiful to see, but it also has a practical utility. And lastly, there's 2 slides I need to show from our PR person from JGI. We do have a user meeting from March. We do talk about a lot of this kind of study happening in JGI. So if you're interested, the registration is open right now. The area we cover will be both technology and scientific talk. And we have a technology workshop, just talk about specific on a lot of methods we do, including epigenetics, transcriptome, so on and so forth. And lastly, we have open CSP call that if you're interested in this kind of project, we're happy to do this for you, as long as you're putting a proposal, your different kind of proposal that actually solicitate this kind of projects. So if you go into the website, you can see we have a lot of proposal call, and just this is the right time to do this. That's all. Thank you.
Thank you very much.